I mean, just you can mute now. Then I will let you know when to unmute and you can start. Yeah?
Yes.
Am I audible?
Yes, you are, Devika.
Okay. Hello, everyone. Hearty welcome to all the participants and eminent speakers to this webinar session organized by the School of Biosciences, Mar Athanasius College for Advanced Studies, Tiruvalla. Every year, our college used to conduct international conferences on trending topics in the field of science. Till now, we have conducted around 80 webinars, and this one is the first international webinar series organized. This online conference is scheduled from 20th May 2021 to 31st May 2021. Today, we are about to start our first session of the international webinar series focusing on the current trends in molecular life sciences. Mar Athanasius College for Advanced Studies, Tiruvalla, MACFAST, is a postgraduate research institute established in 2001, offering the best courses in business management, information technology, and biosciences. The college is affiliated to Mahatma Gandhi University, Kottayam, and approved by the All India Council for Technical Education, New Delhi. On behalf of college, once again, I welcome everyone to this webinar.
Now, let's begin our session with a silent prayer. Okay, thank you. Next, I invite all the dignitaries to inaugurate the international webinar series on current trends in molecular life science. I request Dr. Jenny Jacob, Head of the Department, School of Bioscience, MACFAST, to deliver the welcome speech.
Thank you, Aparna. Hello, welcome to all participants to our international webinar series. So the webinar series, international webinar series is starting from today onwards, that is from the 20th, and it is extending till the 31st of May. So let me enter into my duty. Principal Father Dr. Saryan J. Kottail, Administrator Professor Varghese Abraham, our distinguished speaker for today, Dr. Peter Marhavi, from the Plant Science Center, Sweden, our research director, Dr. Jyotish Kumar Parmeshwaran Pillai, our dean, Professor Beena Cherian, program coordinator, Dr. Sibu Simon, distinguished participants, students, and my dear faculty members. So hello and welcome to all the participants to the international webinar series on current trends in molecular life sciences. It is indeed a glorious moment to extend my warm wishes on behalf of the School of Biosciences and MACFAST. Thank you for taking the time to attend our webinar series. And I would like to let you know that we have been continuously, as it already said, we have been continuously conducting webinars during the lockdown as part of the G.N. Ramendran Science Club, which is affiliated to the Vigyan Prasar Government of India. And fortunately, we have already conducted 82 webinars successfully, thanks to all our participants from different parts of the country. And through conducting a continuous series of webinars, we intend to revive the spirit of science and to spark young minds into the marvelous realm of scientific research. So at the onset, I heartily welcome our beloved principal, who has always been the pillar of support behind all the activities of the School of Biosciences. Welcome, Father Dr. Saryan J. Kottail, to our international webinar series. Professor Varghese Abraham, Administrator, has also been a constant encouragement to us and have extended all the support that is required for the activities of the School of Bioscience. So welcome, sir, to our international webinar series. I wholeheartedly welcome our esteemed guest of honor and speaker for today's session, Dr. Peter Marhavi from the Plant Science Center, Sweden. Thank you, sir, for accepting our invitation and hope we have a fruitful discussion from today's talk. Our research director, Dr. Jyotish Kumar, and our Dean, Professor Beena Cherian. You have been insightful during all these past years. Actually, Research Director has joined us very recently, but our Dean, Professor Beena Cherian, has been with us for a long time, and she has always provided all the insights for us. We welcome you, sir, madam, to this international webinar series. And because the thrust of the whole webinar is the talk that you're eagerly awaiting for, we can go into the talk. But before that, I would just like to say that there is a special welcome to all the directors, HODs, faculty members of MACFAST, and particularly I would like to express my heartfelt gratitude to all the department faculty members for making this event a grand success. And I really believe that this whole process would have been impossible without your support. So thank you one and all. And regarding the participation for the webinar, because we have an overwhelming participation for the webinar, we have decided that we will be going for YouTube live streaming on YouTube. So those who are not able, your friends who are not able to join in this webinar, they can go to the YouTube site also. Okay. Thank you for your participation in the webinar. Thank you.
Thank you, ma'am. And I welcome you to this section. Now, I invite Father Dr. Cherian Kote, Principal MACFAST, to deliver the presidential address.
Yeah, very good evening to everyone. Dear Professor Dr. Peter Marhavi from Sweden, dear directors, HODs, delegates, and my dear students, it has been a great joy and privilege to deliver this inaugural address in the commencement of international webinar series on current trends in molecular life science, organized by the School of Bioscience, MACFAST Tiruvalla. I'm sure that this conference will explore the recent advances in molecular biology and its impact in life sciences. This conference will compile a series of scholarly presentations from eminent scientists and scholars from various prestigious universities around the globe on a single virtual platform. I thank and appreciate the presence and contributions of Professor Dr. Peter Marhavi. I hope this conference will contribute a lot to our society in mitigating COVID-19. I take this opportunity to thank and appreciate the Team Bioscience Department for organizing such a series of webinars on this relevant topic. I thank Professor Varghese Abraham, Administrator, Dr. Jenny Jacob, HOD, Dr. Sibu Simon, the coordinator of this international webinar, Dr. Smita Vijayan and Dr. Teresa Varghese, the members of the coordinating committee. Dear delegates, I wish you all the very best and have a fruitful session. May God bless all. Thank you.
Thank you, Father. I welcome you too to this session. Now, I request Professor Varghese Abraham, the Chief Administrator, MACFAST, to address the audience.
Varghese sir.
Hello, am I audible to you?
Yes, sir. Yes, sir.
A very warm welcome and good afternoon to all. Hope I am audible to all of you present in this webinar.
Yes, sir.
Yes, sir.
Dear loving Father Dr. Cherian Kote, Principal MACFAST, our guest of honor and principal speaker of today, Dr. Peter Marhavi from Sweden, Dr. Jenny Jacob, HOD, School of Biosciences, Dr. Sibu Simon, coordinator of this webinar series, Dr. Jyotikumar Parameswaran, Associate Professor and Assistant Director, MACFAST, Dean, School of Bioscience, Beena Cherian, Associate Coordinators of the webinar series, Dr. Smita Vijayan and Dr. Teresa Varghese, and all my faculty and student colleagues. I'm happy to know that the Research Forum of MACFAST, in association with the School of Bioscience and Accuracy, is organizing an international webinar series during the last 2 weeks of May 2021. My congratulations for the HOD, Dean, School of Biosciences, and for the full team of faculty members of School of Bioscience for their colorful continuation of the tradition of conducting webinar series ever since COVID-19 pandemic paralyzed our life and activities and denied us any kind of direct face-to-face interactions. This year, they have selected a very relevant topic, current trends in molecular life sciences. As we all know, worldwide research activities are going on in this area as molecular life sciences open up opportunities for multidisciplinary involvement, leading to the development of useful solutions to key issues in many areas. It touches areas like food science, cell biology, biosafety, biosecurity, conversion of biodiversity, etc., etc. It is my hope that the eminent professors and researchers participating in this webinar series, which is inaugurated today by our loving principal, Reverend Father J. Kotte, will seriously discuss this and associate and take benefits for all the stakeholders. Once again, I would like to congratulate the team Bioscience for its HOD Jenny Jacob and coordinator Dr. Sibu Simon for providing a wonderful opportunity for our students and faculty members, as well as for all the participants of this webinar. I hope this will give them a fruitful time and initiate their research spirit to do research and come out with useful products for the society. Stay safe, take care. God bless all of you. God bless MACFAST. Thank you.
Thank you, sir, for sharing your thoughts. Now, I invite Dr. Danya Radhakrishnan, Assistant Professor, School of Bioscience, MACFAST, to introduce the speech maker today.
Thank you. Am I audible, Jenny ma'am?
Yes,
Yes,
yes, yes.
So good afternoon to one and all. We all know that plants are constantly subjected to injuries during their lifespan as a result of their sessile nature. Wounded plants are easy targets for parasites and pathogens. So they need to display certain mechanisms to ensure their fitness and survival. On this context, I would like to introduce you to an eminent scientist, Dr. Peter Marhavi, Assistant Professor at the reputed Swedish University of Agricultural Sciences. He will enlighten us in his interesting talk about how wounded plants coordinate their regeneration and defense responses. It is indeed my pleasure to introduce you to Dr. Peter Marhavi. I can confidently say that Dr. Marhavi is counted one among the most promising young PIs of our times, and he has been a successful scientist in the field of plant developmental biology. Dr. Marhavi began his scientific education in Comenius University in Slovakia, from where he obtained his bachelor's and master's degree. He then pursued his PhD from the prestigious VIB Department of Plant Systems Biology at the Ghent University in Belgium. Following this, he acquired a rigorous degree in molecular biology, again from Comenius University. He also has 6 years of postdoctoral experience from University of Lucerne, Switzerland, VIB Ghent, and Institute of Science and Technology, Austria. All through his remarkable research career, he has worked with some of the most distinguished scientists in plant science. Till date, he has published 22 papers and many more are in pipeline. He has a cumulative citation of 1,649, proving the impact and the usefulness of his research, which have been published in some of the most renowned scientific journals. Some of his key findings focus on the dynamics of phytohormones, especially auxin and its transporter during lateral root development. His studies also reveal the developmental significance of crosstalk between phytohormones and the cellular and molecular mechanism in plants to elicit defense response against nematodes. Dr. Marhavi also has several years of teaching experience and has served as reviewer for journals and grant review panels. His scientific expertise have been recognized by numerous fellowships such as FEB's Long-Term Fellowship, EMBO Short-Term Fellowship, and Chem Foundation Award, to name just a few. Indeed, it is an honor to have you as the first speaker in our international webinar series. I can assure you that it is indeed going to be an interesting talk. Before I hand over the platform to the speaker, I request all members of the audience to please mute your mic. Kindly reserve all your questions till the end of the talk. You may, however, put your questions in the chat box. Thank you. Dr. Marhavi, you're welcome to begin.
Thank you very much for a really lovely introduction. And I'm really honored that I can be here with you and present my work, which I was doing lately. I just have to try to share the screen. Okay. Does it work now? Yeah, can you see the screen?
Yeah.
Yes, sir.
Yeah,
Yes.
that's good.
Okay, that's good. So as I was introduced, so I mean, I was having experience in the plant development, also biology in past few years. And I would like to just go briefly through my kind of discoveries, which brought me to my position now at the Department of the Plant and the Forest Biology and Plant Physiology in the Umea. And I would start with the introduction of this slide. I mean, with the people who are familiar working with the plants, that they know, I mean, if the plants are stressed, they react to the stress in various ways. What you see from this particular image here is the seedling of Arabidopsis thaliana, especially the root, under the stress conditions. What you see in this white color, that's the root, and what you see the pink kind of like a pipe inside are the nematodes. That what was introduced that I'm mostly focusing now on is the plant pathogen interactions. So before I start to go more in detail to my presentation, I mean, it's just good to compare a few things with what we have a common in animals and the plants. And that's after fertilization, when the first asymmetric division is forming the zygote. And then if you will go with the development of the embryos, like in this case is the human embryo, then you have the embryonic development, so where you develop all the organs and you build individual inside the uterus, and after the delivering, we are like ready-made humans. Of course, after the years, I mean, we're getting mature and stronger and smarter and so on. But with the plants, what is the difference? The little difference is, okay, after the first division, we have the embryogenesis as well. Then you have a small seedling, but then most of the organs are developed in the plants post-embryonically. So this is the big difference between the animals and the plants, that the development is happening mostly post-embryonically, and it's under the tight control of the plant hormones. Like you have pointed here, like several plant hormones, whether it's auxin, cytokinin, abscisic acid, and so on, that they regulate also all different kind of developmental processes. They don't act at the same time when there is a crosstalk. When plants are in a good conditions and they're perfectly growing, like there is a perfect weather temperature, humidity, and everything, then everything goes like in our lab conditions most of the time, then the plants are happy. But then the plants in the nature, they have to cope with a lot of biotic and abiotic factors. And then they have to recognize these factors or the stressors, and then they have to react to them because they want to always get back to the their happy life or to the happy state. And once they are attacked under, for example, with some pathogens, they're activating their immune or the defense responses, and then the plant try to avoid another secondary infections and try to heal their own, let's say, damaged tissues or damaged organs. But then, of course, I mean, it's depend on the degree of the damage or the degree of the infection, because then not always plant can deal with it. That simply sometimes they just lose their battle and they just die. So there are several influencers, like I mentioned you, there is either abiotic or biotic factors. And in these pictures, what you can see, that what is the most obvious, some herbivores when they are eating the plants. So then, I mean, this is the most obvious for us because that's the above ground tissues. So they're eating some branches or the leaves, and then the plants has to regenerate and to deal with these situations. And the regeneration, I mean, this is the really remarkable thing from the plants, that, I mean, they can really regenerate well. Even if you wound half of the organ, or if you cut the branch, the plants can really deal with it. And it's really depend on what kind of plants is it also. I mean, because if you have a lower species plants, like in here, you have the mosses or the liverworts, then actually they're regenerating fully from the small part, then you can get entire plant. But then in more advanced or the vascular plants, if you do the wounding, like in this case, it's a wounding of the root meristem, the root apical meristem, you chop, then you also get regeneration, but this is tightly controlled by the hormones. And in this case, it's mostly regulated by the auxin and cytokinin. And this is already known for really long decades. I mean, that's already the work was showing this, that auxin and cytokinin, the brain grow in the regeneration in 60s. The plants, if we stay below ground in the root, there is the rich environment, and there the rich environment is either with the positive that can be a friendly, let's say, microbes, or then you have also pathogen. And the plants, they recognizing and they distinguish who is the beneficial and who is the necrotrophic pathogen, and then they're reacting, they're building the machinery of the defense systems, and they're sending the signals through the volatiles even to the neighboring plants. They're building a really defense system to avoid the pathogens as much as possible. Then if you have the degree of the damage, I mean, in the soil, you have a lot of microbes that they want to basically damage the plants. And what you can see from the movie up here, I mean, you have for example, nematode, which is crawling into the Arabidopsis root. I mean, what is the red color you see it is the propidium iodide staining, that is staining the cell walls, that we can visualize the individual cells in the plants, and then you see the penetration of the nematodes. But this is really only one species among only the plant parasitic nematodes is over 4,000 species. And then I don't mentioning even other bugs you have it here, or you have the fungi, or you can have microbes, and so on and so on. And they all somehow also invade the plants in a different way. You don't in this case, the nematode, when they go, they're breaking the cells. They go from outside of the soil, I mean, from the environment, they're reaching into the plant, and they break cell by cell until they're reaching their favorite position within the root, and they start to feeding and establishing the feeding site. But then you have another, let's say, different bugs that they don't break the cells, but they slide their stylet or some kind of device. They want to suck out some nutrition out of the plant, so they're sliding between the cells. So you don't break the cells, so then it will just go through the apoplastic space until the region where they want to go, for example, to phloem and to suck out some kind of sugars. Just for your better introduction, or let's say to show you the Arabidopsis root, because I'm working mostly on the Arabidopsis root, which is the model organism for me, and it's have several reasons why do we work with the Arabidopsis, because the root is very simple way structured. I mean, you have it individual cell files, what you can see over here. So this is first the seedling, let's say 8 or 9 days Arabidopsis seedling. That you have the primary root, you have lateral roots, and you have a cotyledons and the leaves. Then this is the image from the region over here, what you can see the red square. And then you have the confocal image from the fluorescent microscope or confocal fluorescent microscopy. You stain your roots with the fluorescent dyes, or you can have it like even the genetically encoded fluorescent markers that they visualizing the cell shapes. And then this is the cross sections from this region where you can really see how the root look like. If you just cut it like transversally, you go from one side to the other side. And just to show you briefly kind of like anatomy, you have like epidermal cell layer, which is the outermost cell layers, below is the cortex. Then you have endodermis, and which is closing the vascular cylinder that belongs there, like a pericycle and a xylem, phloem, and so on. Why I'm showing you this? Because if you want to study the plant responses, in this case, you can do it as I showed you before. I mean, you can do kind of like damage of the plants, which is a bit more drastic or more progressive. That means that you damage like a lot of cells. But in my research, we are focusing mostly on the individual cell that you can do damage and how you're going to do it. So now, I mean, there are the new ways or the new tools that we can really particularly damage one cell inside the plant root, what you see from the movie up here. And then you have a cross sections down here. What you see from this movie, it's a laser ablation system where with the microscope you can set the position of the laser accumulation in the cell that you can kill one individual cell which is deep inside the tissue without damaging all other tissues over here. So this is just the movie to show you how does it work. And then the root is stained again with the propidium iodide, which is this apoplastic dye. And then once the cell is dead, you see this penetration of the dye going inside the plant roots and which are the damaged. So this is like a proof of concept, like, okay, if there is the red color very simply set inside the cell, then the cell is dead, and what you can see over here. Then if you look at the image first on the left side over here, this is the before ablation. So you see there is no any white color inside. And this is after ablation, where we ablated the endodermal cell. That what you can appreciate from the images, that there is no penetration of any dye somewhere above or below. So you can really specifically aim for whatever cell you like in the tissue and to kill it. Yes? Is there any question or? Okay, so I think that's not for me then. Then
yes,
we can
I
go
can
on.
proceed.
Yes, and then I mean the laser ablation was not really invented by me. That was already shown that the system is working in 90s. And then what they were doing, they were focusing mostly on the root meristem, so which is the lower part of the root, where is like active cell divisions. And then they were using ablation to see like if the plants or how the plants are dealing with this process. And they were showing, okay, there is certain regeneration that you can get, like they can study the cell fate or the cell differentiation. Later on, what we tried to do, so we did, okay, a bit more investigation. What's happened if we go into the root meristem, so which is the region over here. This is this very simple schematic. And then if you ablate one single cell inside the root, and then what we find out that if you ablate the cell, you always stimulate the cell division of the cell which is below or the next to it, to the ablated one, but always go from the inside towards the outside. So you stimulate the division. For example, if we ablate the cortex cell, then we always stimulate the cell division of the endodermis, which is also keeping the cell fate of the endodermis. And this was shown like a few years back. I mean, what you can see the citation over here. And here you have the actual images that, I mean, if you ablate the cell over here, then you stimulate these divisions, you're getting like a replacement of the tissue. Another report, they also showed that this regeneration of the processes with the laser ablation, it's actually important player in this process is the jasmonic acid, which is acting in this respect that it's stimulating somehow the regeneration and the stem cells niche propagation. Then we were also curious what's happened actually in the other parts of the roots, because if we are here in the very root tip of the root, where you have the meristematic region, then the cells are still actively dividing. But then they stop to divide somewhere here after the elongation zone. The cells, they just elongate and they do not divide anymore, except of the one tissue, which is the pericycle, because the pericycle is giving the rise of the lateral roots, that what you're having over here. So we were interesting, like what is happening in that stage. But before I will show you the more details, I mean, this is how the lateral roots start to divide. This is for your imagination. If you have a seedlings like this, you go again inside the deep of the root, and then you have a pericycle cells that I told you. This is the part of the vascular system, and this is the unique feature of the pericycle cells, that they can divide also in the other part of the roots, and they're stimulating the division of the lateral root primordia until it's forming the lateral root, what you can follow up from this here, from the video over here. What is the main advantage actually if somebody is studying the lateral roots? I mean, you can see it obviously that you can study it from the beginning of their development, which is over here, that you can study the divisions, the orientation of divisions. I mean, you can do whatever you can imagine with that stuff, but also what is also good, that you have not only one lateral root like the primary root, but you have a lot of lateral roots, and they constantly dividing until this plant adult age, and then until it's reaching its stage, and then it's just growing out of the maternal tissue, what is just demonstrated over here, over there. But then I also mentioned you that the lateral roots, they are also regulated by the different hormones, and then is mostly under the action of auxin and cytokinin. While auxin is acting positively on the development or the initiation of the lateral roots, cytokinin has the opposite effect, is mostly acting as a negatively, or is suppressing the either division or the outgrowth of the lateral roots. Another very important tissue during the lateral root initiation or the formation is the endodermis, which is the one cellar above. Because what we find out previously, that before you start to accumulating auxin in the pericycle cells, then which they turn to the founder cell, and then you also getting localized, very localized expression above those cells in the endodermis, which is transient. Then another report showed indeed that the endodermis is very important for the stimulation of the lateral root initiation, because if you overexpress like dominant negative SHI2, so for example you block or you negatively influence the auxin signaling in endodermis, you end up with the roots which are completely naked, you don't have any lateral root initiation. So our question was like, okay, whether we can override the system. So what we did, so we took the laser ablation approach, and then we ablated the endodermal cells along the axis of the root in the different positions. What we find out that if we ablate the endodermal cell, then we activate the cell divisions, what you can follow up here. What you see, this is the microtubule marker line, MAP4, which I used to track also the lateral roots and the cell division, because you have the spindle formation, and then you see that how the division is happening, and also you can study the orientation of the cell division. So, but this was really only the unique feature of the endodermis. So if you release the constraint from endodermis, which is pressing towards the pericycle, then you activate the cell division even without the auxin. And then we tried the same experiments with the different cell tissues. I mean, whether is it the cortex or epidermis, none of them lead to the same response as the endodermis. So we concluded that endodermis is really acting as a constraint between the endodermis and pericycle. So it's imagine is like a pipe within the pipe. If it's released, then actually the other one can expand. And this was what was happening. Just for your illustration to see a bit better, again, if you have a normal any ablation, this is the control situation, you see that the lateral roots, they start to dividing in the anticoinal divisions. So you have one, second, another division, and then after a while they change the orientation plane and they start to form the second stage, and then they start to divide the periclinally. So what you can see over here, and the movies are running, let's say, like overnight. Then what we did in the one movie below, what you can see over here, we did the ablation of endodermal cell. And what we were just waiting, we were happening like what's going on. You see then you're getting suddenly again division. But what was really surprising, that this division was directly going like to the second stage. So except of these first anticoinal divisions, you suddenly start to jump and you override the system and you go into the periclinal divisions. Then what we tried, I'm not going to show the data, but you can see it on my manuscript if somebody is interested. When you apply the auxin, because auxin is apparently very important for the lateral root initiation, then if you do even the ablation, you stimulate these divisions, but then you form a normal lateral roots with the normal order of the divisions. So they start to go again anticlinally, and then they go to the periclinally. So to conclude this little part here, what we can say that the endodermis is really crucial for the initiation processes of the pericycle. And the auxin is very important for orientation of the cell division and the organ formation. Then we were also a bit interesting because, I mean, if we are doing this wounding or damages on the cellular resolution, then I was also curious, okay, is there any activation of the defense responses or how the plant is actually recognizing, because this is not very common that there is any lasers in the nature. And this is, I mean, you can say it's kind of artificial. Then we were interesting, like, okay, can we actually see some defense response in these regards? And how would the cells will respond to this reaction? So what we find out, or I mean, before we decided what we find out, how the animals react, I mean, to the situation. We have a brain, or we have the central nervous system, right, where we have the neurons and we have a chemical communication through the nerves, and then we know that if something is going wrong or good, we reacting to it. I mean, we can react in either positive or negative way. Like, for example, in this, the lion is chasing a herd of buffaloes, so they see that there is a reaction, so they process the reaction, they process the input, and they react, and they start to run. But then it can be also the situation, but then maybe they change their mind, and instead of running away, they start to run towards the intruder, and they try to chase him away. Plants, they do not have a central nervous system, but then do pose with a lot of chemicals, and the chemicals are serving for the plants as a messengers. And in this little scheme over here, I will just give you a brief overview that if you have a plant and then the plant is damaged, then plant it's having reactions via the electric or hydraulic signals that it's actually going from one leaf even to the other leaf. So it's a systemic signal propagation. Then you have also like reactive oxygen species activation. You have a calcium spike or the calcium wave propagation, which is leading to the final output like upregulation of the hormones that they act in this particular process as a defense mechanisms against the intruders. And in the aerial tissues, the most of the time, the hormones they are acting as a defense compounds are either jasmonate, salicylic acid, ethylene, or the ABA. Then how is this long distance communication happening? And as I told you, you have the hydraulic or electric signal. And this is known already between many plant species, and it was already described also when you have carnivorous plants, and then the fly it's coming and it's sticked to the sticky tentacles, then the plant, or we know that they are either closing or they basically trying to trap the animals inside and to digest it and to use it for the nutrition. And then it was recorded that actually this is really purely electrical signal that actually is a stimuli, and then the plant is reacting. What was also shown that this actually it's very long distance signaling, because it can go from one plant to the from one leaf to the another one, or it can propagate throughout the plant. I mean, this was all done above ground. But the concern for me was like, okay, if we do the ablation in the root, can we see any electrical signal propagation or any defense responses and so on? But because so far there was like a minimum reports done on the root defense responses in general, and I think the main reason for this because I mean it's easier to work with the leaves or above ground tissues. What is also known, I just jump too far, but I will go back. So if you have this signal propagation, they already know also the molecular mechanisms behind the electrical signal propagation in the above ground tissues. And what was shown that is actually the glutamic acid or the glutamate, which is released after the wounding by the carnivorous plants or by the mechanical damage, which is basically transmitting this signal, and also it's regulating the calcium wave propagation, which is then actually either stimulating or inhibiting the response of the hormones. And that was shown by the really beautiful paper Untitled Chapter 2018. And at the same time, they also showed that actually the electrical signals are really also regulated by these GLRs. So then actually the glutamic acid, at least in the above ground tissues, is acting as a key regulator for the propagation of electrical signal and the calcium and the responses in immunity build. But then how is the situation in the root? As I told you before, I mean, there is almost no information or very little about the root plant defense responses, and there was no information about the electrophysiology whatsoever that time. So what we did, we just designed the experiment in a way that where we put the electrode, it's a silver wire electrode you're placing on the root surface. The better image is actually maybe over here down in the corner, where we put gently on the epidermal cell layer of the root, you're putting the electrode, and then we're doing the wounding of the single cell somewhere here on the other side. We always put the electrode bit further away, we shift it to the left or the right side, that you don't have a direct interference of the objective of the laser with the electrode, that you don't get the false positive signals. And we were doing the ablations of the cortex cells. And what we find out actually was quite striking, because in the if I will jump back here, if you will have the electrophysiology recording over here, and you do the smashing of the leaf, which you imagine that you smash like a lot of cells at the same time, you're getting the depolarizations around 100 millivolts. And then with the single cell, actually we got almost similar value and in the root. So then actually what we can say that in the root, even if you damage only single cell, you're getting these really big depolarization peaks. So then the plants have an active electric signaling also in the root. And then we were also curious, okay, does this propagate for the long distance, or this is rather like a localized response, or is a short distances? Because as I told you before, in the plants, I mean, if you have the leaves and you smashed this leaf over here and you go in the centimeters, then the signal is propagating to the other leaf and so on. And this was reported already several times, that it's like really rapid propagation of the signal. But then how is it in the root? So what we did the experiment, we placed the electrode in the different positions, and we did the ablations. And what we find out that if we do this ablation, then we get the depolarization really strong, which is relatively close to the wounded part. But then if you go a bit further, I mean on the root, if you go like 200-300 micrometer away, you hardly detect any signal. So that was for us conclusion that okay, in the shoot and the root, they do have an active electric signal, but the propagation is really different. While in the shoot you have it mostly systemic signal, in the root, at least for the electrophysiology recording, we do have mostly the localized or cell-to-cell responses, in which they don't go so far. Then, as I told you before, I mean, if you do the damage, you don't activate only the active electric signal, but you also stimulate the ROS production and the calcium. And what was already shown also in the root, that if you applied the drop of the salt, like 100 millimolar NaCl, then you see the calcium responses over here. What you see, you have a genetically encoded reporter marker lines, or you have also like a thread-based systems, where actually you can see the responses of the calcium, and you see the travel. And then they also saw that, okay, if we apply the salt, then we have the propagation through almost the whole root. I mean, because you see that, for example, they place it over here in the root tip, and they saw that it's really traveling a bit far away from the applied area, which is fine. But what we did, again, we are not, or what they did, I mean, if you imagine that if you put 100 micrometer salt, microliter of the salt, then you're actually covering a lot of cells. So of course you're getting like a huge burst or the way. But what we did, it was a slightly different. We took the genetically encoded sensor, which is in this case RGECO, but you can have also like a GCAMP, or you can have a Case12. There is like many of them available. And what we did, we ran the movie and we ablate the cell over here where you have the arrow. And what we saw that after ablation, you immediately getting this really huge wave of the calcium. But what we saw it also, that this is also relatively localized. It doesn't really go much further. So it started to really propagate. I will just play it again. it starts immediately here in this cell filing cortex, then it's going like inside. And then you would see also somehow like the wave is traveling from cell to cell. Why this is like switching it off, then it's switching it on in other ones, and then it would stop somewhere here. And then when we did the quantifications of that, you would see that after the ablation, you're getting like the maximum peak, it's approximately like in 7 seconds after. And when you use some inhibitors for the like a calcium channels, then you see that you basically, you don't block it fully, but you reduce a lot the calcium wave propagation. So to conclude this part, I mean, we find out that actually it has to be actual damage. It cannot be that you just basically tangle the cell somehow, because I will show it to you why. If we set the laser in a position that you do not kill the cell, but you just somehow put a bit of heat on the cell, that you just somehow like pre-warm it, then you get the response in this one single cell, but the wave, it doesn't propagate. So that was conclusion for us, okay, that if we do this, I mean, what the calcium wave is really propagated is only when you do the actual damage and not if you just pre-warm the system. Then, and if I would be concluding these 2 little parts from the story, so what I showed you so far, that we have the active electric signal in the plant roots, which is localized, and at the same case, we have also the calcium, which is again very localized if you do the minimum damage. Then the main question was like, okay, what would be the primary defense response? Because in the shoot, I show you the plants are responding mostly via the jasmonate signaling if you do the wounding, or you activate also the abscisic acid, or you can be also having there the salicylic acid. And this is like if you do the either manual wounding, or you can use the carnivorous caterpillar, not carnivorous, sorry, the caterpillar which are chewing the leaves, and then you're getting the same responses. Like the defense of the plant is activating the jasmonate. But that was the case for us or not. So actually what we tried, so we have a jasmonate reporter marker line that is tagged with NLS YFP, so it should shine in the green. And then we run the time lapses, or we check the plants. In the control situation, we don't see any fluorescence coming up. And then if we did the ablation of the single cell of the cortex, we also neither see any accumulation. So then you have 2 possibilities. First one, your construct is wrong, or second one, the plant doesn't react the same way in the root like in the shoot towards the pathogens. And then another confirmation for us was that actually the second scenario is correct, because then when we took the cotyledons or the leaves of the Arabidopsis plant, we stained it again with the propidium iodide, and when we ran the movie, these movies are overnight, then you don't see any fluorescence coming up in a green, yellow, or yellow nucleus. But then when we ablated the cell, which is the ablated cell over here, you would see that actually the neighboring cells, they start to activating the response, the defense response, and activate the jasmonate. So to conclude it, again, I mean, as I'm saying, what we find is not only that we have the active electric signal, calcium vein, which are localized, but also we do have a activation of the immune responses, which seems to be slightly differ between the shoot and the root, because if we applied the cell from the same construct in the root, we don't get any jasmonate, but if we do ablate the cells in the shoot, let's say in the leaves, then we activate the signal, and then you're getting the responses. Then the question for us was, okay, so what is it? What is the next possible defense marker or the mechanism for us? And then we came to ethylene, because ethylene is already known for a long time. It is acting as a defense compound, and also for the wounding. If you do like a mechanical breakage of the plants, then actually you activate a lot of ethylene. So what we did, so we have the ACA6, which we use as an ethylene reporter marker line. And if we run the time lapse, let's say for 5 hours on the confocal microscope, then you see that already in 5 hours in the control situation, you get some kind of response. You see the blue yellowish nucleus coming up. But then after ablation of the cell, which is marked over here of the single cell, we getting really much more increase of the intensity, and these are the quantifications. So what we can conclude also from this, compared to the shoot, that when we activate with the single cell damage, we in the root, we activating mostly ethylene, but not the jasmonate. Then, of course, your obvious question would be, how is this really relevant? That's what I already said in the beginning, that, okay, in the nature, there is not really many lasers. And then we are trying to find the best optimal system, because what you can see from the laser ablation is that actually the cell that is happening very fast and is sudden. So you cannot use the bacteria, for example, which they digesting the cell wall or all these kind of directions. But also we need to have a system that is basically breaking a single cell at the time. And then we came up with the nematodes, because the nematodes, you saw already the movie before, and there is another one, they go through the root and they're breaking cell by cell. And this is also relatively fast. So actually when they're moving the stylet, which is this structure over here, they basically checking the cells, and then once they decide, okay, this is the correct cell for me, then they just break it and they go through, and then they break another one cells. So actually we were thinking that the nematodes, they can actually mimic, or I mimic, or we mimic the nematode with the laser, and we were curious whether we will get the same responses of the plants. The problem with the nematodes, I mean, is a little bit that they are live animals, right? I mean, so to work with them is a little bit more difficult, like if you have a super controlled conditions with the lasers, because then you just grow your plants and you can just go and you can use I mean, whatever you want to do. But with them, I mean, you have to do the infections, and not always they want to attack the root, or they just attack the root in a way that it's out of your field of view and so on. And here is just the representation of the different species of the nematodes. So the nematodes I'm presenting here are the cis nematodes, that they are just going really outside from they are born outside, and then they go inside the root. I mean, they find the position. They don't have a really defined spot if they prefer this region, or that region. They just go somehow randomly. We don't know. There is research going on that. I mean, like how they choosing the cells, but we are still far from knowing it. And then they just decide, and then they go and break the cells, and then they want to go inside the vascular system where they're establishing their feeding site. And then they start their parasitic life in there. So what we did, we took our reporter marker lines, and I mean, also I'm not going to show you here because I don't think we have much time for that. But we find out that if we put the nematodes on the jasmonate reporter marker lines and they do the damage, there was also not really response. So what we did then, we placed also the nematodes on the ethylene reporter marker line. What you would see here, I mean, it's above, it's a control situation, and down it will be when the nematode is invading. So you see the nematode, it's invading the root. So what is in this red circle, and this is belonging to the DIC image. So you see this huge increase of the ethylene response, while in the control situation, you get some response because obviously the plant is a little bit stressed if you're scanning it on the microscope for like 15, 16 hours. But then you're getting this really huge activation of the defense signal. So now the question was, okay, could we really also detect the depolarization or the surface potential changes with the nematodes, or it's just really that we were lucky with the laser ablation? So we kind of challenged ourselves, and I placed the electrode on the roots. So this is the actual experiment. You have a control, and here is with the nematode. So where you have these black spots, there the electrode is placed, and then, I mean, in the control situation, if you do the recording, so this is the small part of the recording, then you don't have any like a depolarization of the shivering. But then once the nematodes go relatively close to the electrode and they cause the damage, like for example, you will see it very soon in this situation when they are already here, then you started to getting these depolarizations, so which is kind of the same way like we saw it with our laser. And then if there is just like a massive destruction of the root, then you're just getting, pure jumping up and down the electrophysiological recording. And this was the quantifications. The quantifications show that, okay, it's like half reduced compared to the laser ablation, because that can be, that was what was recorded. And then we still get the depolarization, but again, as I'm saying, it was not going far. I mean, the damage has to be relatively close to the placed electrode to be able to detect these depolarizations. So to kind of conclude my talk from this part was like what we shown, that if you use the new tools like a laser ablation, you can mimic the natural situation that like parasites are acting on the plants and how the plants are reacting to it. And in this case, is the plant after wounding is stimulating or is putting like you're getting the surface potential changes, you're getting ROS and the calcium wave, which activating then the immune response in ethylene, and that one is acting as a putting away the nematodes and they don't want it. And then I'm coming to my last slide, where I would like to thank few people. I mean, that's now currently my group, which I have it here in the Umea, and also to my past places where I was spending my postdoc or the PhD. And of course, I mean, the grant agencies which are shown down over here that I got the grant to perform the research in these directions. And again, I would like to thank you, the organizers and all of you, for your attentions and for inviting me and to giving me the chance to present the work. So thank you very much.
Thank you, sir. It was a great presentation. So thank you so much for accepting our invitation to come and present at School of Biosciences.
Thank you. So now I can stop sharing the screen, and if there are any questions, I would be happy to answer.
Thank you. I'll be taking questions from now onwards. So does anybody have any question that needs to be asked to the...
There are questions in the chat box, and you can also raise your hands. So,
Dr. Marhavi, I'll start with a question that has come in the chat box. So we have a question from Dr. Sini, who asked which gas is used for laser ablation?
Which gases?
Basically, I think she's asking what is the method of inducing laser ablation?
Well, I mean, you have you need to have either 2-photon microscope or the confocal microscope with the UV laser coupled. I was using both. How the system works is basically if you have a UV laser coupled with the confocal microscope, it's like a kind of cold killing. So you just go very fast and that's it. And you are basically accumulating the light in a certain spot in the cell, and then how the cell expand. The second method, which I'm using currently, is the 2-photon laser ablation. So where you have to set or to optimize the condition of the laser, where you accumulating actually not really even the light, but the heat, which is actually killing then the cell. So you need to have a 2-photon or the confocal microscope with the UV laser, not 405. You need to have a really UV laser, if I answer your questions.
Okay, that I guess answers the question. Do we have any questions from the audience? You may raise your hand. Okay, we have one more question in the chat box from Sumera, who asks, does the minerals level in the soil also affect the laser ablation?
That's a good question. I would love to know. I mean, we're going to do something in this direction because at the moment we done everything only on the medium, which is solidified with the agar. So we didn't do actual experiments with the soil. So I cannot answer for that question. I believe there will be some interference because, I mean, if you imagine the minerals, like after the ablation, I mean, you're releasing the contents of the cell, right? I mean, that can be like salt, or it can be like all sorts of stuff. So of course there will be some influence. But I cannot answer like what would be the influence.
So, is there any question? Okay, so then I'll ask a question. It's about, is there someone?
Yeah, I will ask. Can I ask a question?
Yes, sir. Yes, sir. Please.
Okay, Peter, excellent talk. Yeah, thank you so much for such a nice talk.
Thank you.
So my question is, so initially you wounded the plant with a mechanical signal, then you validated it with a biological agent. And to some extent, you are showing that you got a similar signal. But what I want to ask you that how confidently you can say that the signal, the wound generated by the mechanical agent or the biological agent, it's how similar they are. How confidently you can say that they are similar signal?
I mean, what I can say is that basically whatever we tried, I mean, whether we try to study the calcium response, electrophysiology, or the immune responses in a way that it's either jasmonate or ethylene, it always reacts the same way. Either it was with nematode or with the laser ablation. If we use the inhibitors like chemicals, I mean, this is a bit tricky with the nematodes because, I mean, if you add some, like toxic compounds, they don't like it. So, I mean, this we cannot really, let's say, put it in the same context. But whatever we tried, it was working or reacting the same way. What we also try, actually, what we are doing now currently in the lab, I didn't present this data because, I mean, that's ongoing work and we have to accumulate it more. But what I can say now, if we use the nematode, or the laser ablation, we also induce the zuberin reaction or the lignification or also the callose deposition, which is also actually happening with the normal natural situation. If you just break the cell, break the organs, or if you even use the nematodes. So actually it's also doing in this direction. So far we have all the leads going that actually we can really, at least in this respect, mimic the nematodes.
Okay, excellent.
With the laser.
Thank you. Excellent. Thank you.
We have one more question from the audience. Remya asks, what is the duration of laser ablation done to the cells, both for root and shoot?
So for the duration, I mean, that's very fast. I mean, normally what we do is it's like below 1.5 seconds is the ablation, or let's say the actual laser pulse on the cells, and it doesn't matter whether is it actually in the leaves is even shorter, because then, I mean, you are on the surface, so it's just like, I would say, between 0.5 second to 1.5 seconds for both systems. That's sufficient.
Anybody having a question may raise your hand. Dr. Peter, I have a question. When the wound signals are induced, is it species specific? Like, is it species specific to the nematode, the wound signals that are induced?
Not really. I mean, what we did is I was using only Arabidopsis for studying the plant immune responses. But in the paper where I showed you that what we were trying to do the laser ablation on the regeneration processes of the root meristem, when the cells are dividing, towards like outer tissue, there we tried to also ablate that was a rice, tobacco, and they were all reacting in the same way. So I don't think this is like a plant species specific. Of course, what I can say that, as I mentioned, if you have a nematodes, there is like over 4,000 parasitic species only for the plants, and they have a little bit different way how they damage the cells, or how, and then the responses can be slightly different. And what I can give you like two example. What I presented here is a cyst nematodes, so they break the cells. But then we also work a little bit on the root knock nematodes, and these nematodes, they go into the root in a different way. They don't break the cells, but they slide between the cells. So, but I cannot tell you the responses now, because this is now ongoing, and I mean, we do not really know. I mean, but we believe that the responses would be really different, because also what you showed from the movie, when we did the ablation, you stimulate or you activate the calcium wave propagation. But if you don't kill the cell, you just basically, hit it a bit. It just activate in one cell, but not in the neighboring cells. So that's why I think it will be slightly different. So your question or my answer would be yes and no.
Thank you. We have one more question from the audience. Deepti asks, which phase of healing is responsible for regeneration?
Sorry, I didn't really understand. Could you repeat?
Which phase of healing is responsible for regeneration? I guess she might be asking how the regeneration process begins after the wounding process.
So it depends where you are in which position of the root. So if you are in this meristematic region, the regeneration or the healing starts that basically activating the cell below, which basically replacing the tissue in a way. So this would be the one scenario. And the second scenario is, as I mentioned it now, that if you go, let's say, to the mature part of the root, and there is no anymore, let's say, cell division going on. So if you do the damage, or if you do the infections, let's say there is mechanical disruption, you're activating a lot of zuberin accumulation around the wound. And the zuberin, we know that is acting as a cork, right, in a way. So actually that, I think, well, how is it working? So it's trying to seal the gap around And actually it's not even for this primary infection, but I believe it's mostly for the secondary infection that actually, they can get like some secondary pathogens coming up. So you have these kind of processes.
Okay. And we have a question from Dr. Reena, who asks, what's the role of microorganisms in plants on changing climatic conditions today?
I not really fully understand if I understand the question, but I mean, of course, now, I mean, I can say it mostly for the Europe, or because now I'm located in Sweden, and the climate is of course influencing the microbial population everywhere, right? So then actually what we are a bit worried now, I mean, here in the Sweden, is that, I mean, you have currently present also the nematodes here, but then the new species can be delivered. And then the very good example could be, for example, there is a pine wilt nematode, which is destroying the entire forest or the trees, which was, let's say, originally in Asia or in United States. But now we start to having this spread also in the Spain because they like somehow the warmer climate. And these nematodes, they're using actually how they transmit, or how they can get also into the northern part of the Europe, is they're using the beetles as the vector for their life cycle. So then basically when the beetle is coming here, for example, it will sit on some pine tree and it will infect the tree, then actually you can start to having problem with the new species which were not currently present. So of course this is a big threat, which I think it will come at some point everywhere because of the global warming. So to answer your question, yeah, I think there will be new species, but we have to fight it in a way, like,
Thank you. I guess students have now become more active in asking questions. We have some questions from students. One of them asks, which cell type plays an important role in regeneration and wound healing due to retention of meristematic ability?
So in Meristem, there doesn't matter where you are It doesn't matter if you ablate the endodermal or cortex cell, or if you ablate the pericycle cell. So it's always the cell layer below which divides actually is trying to fit. What is important to say that if you go behind the elongation zone, so if you know how the root meristem look like, so you go behind and there you do the ablations, you don't get any cell division except of I showed you if you ablate the endodermal cells. So this is the two differences. I hope I answered the question.
And there's another question from Ramya S, who asks, is the response same if any organism other than nematode is acting on the plant?
Yes, I mean, I mean, I didn't try the different, let's say, microorganisms or anything like that. So it's difficult to say, but I believe it will be slightly different with the different pathogens. But if you do, this like sudden fast damage, then it will be reacting the same way. And it doesn't matter whether you are in the closer to the, root tip, or you are basically going far away. So, or if you ablate or if you damage the lateral roots, I think it will be the same response. We don't have a data for that, but this is just my assumption. I mean, what is also shown, what is slightly be different, for the regeneration processes, I told you, if you are in the meristematic region, you're activating the jasmonate under the control of the jasmonate signaling. But, and that was also shown that if they apply the nematodes that they are going in that region, then actually you activating the jasmonate. With our system, if you are not, because these nematodes, they do not go to the root meristem, they always stay in the mature part of the root. So we always see only the ethylene. So what could well be, and this is actually what we investigating one of the postdoc in the lab, is that actually we are having different regions of the root, and we're checking the responses towards the different nematodes and the laser ablations, and we want to try the connection. How is the cross-link, how the basically, or maybe there is like ethylene is upregulating the jasmonate or vice versa, or there is like some kind of like a middleman which is basically activating for both of them, and it's depend on the position of the root or the region where you are placed.
There is a question. Basically, it kind of addresses the overall gist of the presentation. Sri Lakshmi asks, what do plants use to repair their damaged parts? I guess she is addressing the mechanism, overall mechanism maybe.
Well, what they do, I mean, what is shown, I mean, it's that they activate. This is published, I mean, already in 2000s. They start to lignify heavily the tissue and also putting the zuberin, as I said. But exactly the molecular players are still not really full known. So I mean, this I cannot answer.
That is still being studied. Okay. And Dr. Shari Nair asks, what did plants have? Do plants have an immune system?
Yes, that's what I showed. That I mean, it's like activation via the plant hormones, basically.
Yes. So that's the end of the questions in the chat box. I guess that is the end of it. I hand over the platform to Dr. Smita for the vote of thanks.
Thank you, Tanya. Thank you. So a warm good evening to one and all. Respected Principal, Father Dr. Cherianji Kotel, our Administrator, Professor Varghese Abraham, Head of the Department, Dr. Jenny Jacob, Program Coordinator, Dr. Sibu Simon, our Research Director, Dr. Jyotish Kumar, Dean of Science, Professor Beena Cherian, all the participants, all delegates, and my dear students and colleagues. Actually, I'm with immense pleasure, I'm here to offer the vote of thanks because I'm overwhelmed with the participation and also the marvelous presentation by our eminent speaker. So we are very grateful to you, sir, for your marvelous presentation, knowledgeful presentation, and also your awesome interaction with the students. On behalf of the management and on behalf of the programming committee, I express my sincere thanks and thank you, sir. Next, I extend my sincere gratitude to our principal, Father Dr. Cherian J. Kotel, for his constant support, encouragement, and guidance throughout the program. Thank you, Father. I also extend my gratitude to Professor Varghese Abraham for all his support. Thank you, sir. On this occasion, I am extremely thankful to the head of the department, Dr. Jenny Jacob, for her valuable suggestions, support, and help. Thank you, ma'am. I am particularly grateful to Dr. Sibu Simon, Program Coordinator of the webinar series, for his excellent organization and hard work. His efforts must be appreciated specially. Thank you so much, sir. I would also like to thank our research director, Dr. Jyotish Kumar sir, and Dean Professor Beena Cherian for their constant support and encouragement. Thank you, sir. Thank you, ma'am. I also owe my sincere thanks to my colleagues for their kind words and support. Thank you all. And without the participants, this event will not be a successful one. So I express my gratitude and happiness towards all the participants for their active participation, and your presence make this event a successful one. Thank you all. Finally, yet importantly, I have to I extend my gratitude and love to my beloved students for their enthusiasm, positive spirit, and effective participation. Thank you. Once again, thank you all for this wonderful evening. Have a great day.
Thank you, ma'am. So tomorrow our session will begin by 3 p.m. and the link will be given. Thank you.
Thank you, Dr. Peter.
Thank you very much. Thank you and have a nice day. Bye.
Thank you, sir.
Thank you, Dr. Peter.
Thank you, sir.